What is the official definition of a virus? Well, it is a submicroscopic particle consisting of a single nucleic acid, and nucleic acids, remember, are DNA or RNA, surrounded by a protein coat and capable of replication only within the living cells of bacteria, animals, humans, or plants. So please keep in mind that animals and humans are not the only things that can get infected with viruses. Bacteria and plants can also get infected by viruses. So how are viruses transmitted? How are they spread? Well, they can be spread through the respiratory tract, and that's typically how you will get the flu and cold. They can be blood-borne. They can be through sexual contact. They can be through close contact with an animal, or they can be from the bite of an insect, or even the bite of an animal. So viruses are infectious, and infectious means that it can spread from person to person. A non-infectious disease means that it cannot spread from person to person. For example, genetic diseases or cancer. So a couple of terms that you need to understand when talking about the spread of viruses is a pandemic versus an epidemic. And sometimes you'll hear about this on the news or read about it in a newspaper. But an epidemic is when an area is affected by a high number of sicknesses from a specific pathogen. So it's limited to a specific area. So, for example, we had an epidemic this year with the flu. There was a high number of people that got the flu this year in our area. However, a pandemic will affect people worldwide in extremely high numbers. Okay, it's something that's spreading, and it spreads quickly, and it affects a lot of people. And a good example of this is HIV. HIV used to be an epidemic in Africa, so it was limited to Africa. And they believe that it mutated from a monkey. And in Africa, a lot of times people might eat the brains of monkeys kind of as ritualistic. But now, because of global travel, and the ease of global travel and people going to places that they have never gone before, HIV has now become a pandemic, and it is affecting people worldwide. You also need to understand that viruses are specific, okay? It's called specificity, and it means that a specific virus targets a specific type of cell. For example, the HIV virus specifically affects immune cells. And the cold virus specifically affects respiratory cells. That's why when you get a cold, it affects your nasal cavity, it affects your lungs, okay? That's all part of your respiratory system, and those are the cells that the cold virus is attacking. Why they are specific is because the protein coat on the outside of the virus, as you can see here, will match up to the proteins on the outside of those cells. Okay, so as you can see here, it matches up perfectly. So that specific virus will target that type of specific cells, the cells with the proteins that match the virus's protein coat. Very brief structure. All viruses have a nucleic acid or genetic material, which again can be DNA or RNA, and is surrounded by a protein coat. The nucleic acid can mutate, and a perfect example of this is the cold virus and the flu virus, influenza. Each year, that genetic material mutates a little bit every year, and that's why we get a new cold every cold season. The protein coat provides protection, and it provides a vehicle for transmission. So again, remember that the protein coat is what matches up to the proteins on the cell and allows it to get in. Some treatments for viruses. Not very long ago, antivirals started being used. I want to say in the last 10-15 years they really became popular. But they basically, the main mechanism how they work is they prevent the virus from even getting into the cell. Okay, they block the protein. They block the match of the protein so that there is no viral cell recognition. There's also vaccines, and this helps the body get a weakened form of the virus and helps your body build immune defenses before becoming infected, so that when a person becomes infected, they already have the immunities to fight off that infection. So more on viral structure. Something you need to know is that viruses are not cells. Okay, cells are made up of macromolecules. Remember, proteins, carbohydrates, lipids, nucleic acids. And so are viruses, but that does not mean that they are living. Okay? All cells have ribosomes. Viruses don't have ribosomes. All cells have cytoplasm. Viruses don't have cytoplasm. Okay, etc. and so forth. They can't function on their own outside of a living thing. So, the two macromolecules that viruses do have, nucleic acids, which I've mentioned, and proteins, which I've already mentioned. They don't have organelles, they don't have a cell membrane, they don't have cytoplasm. But viruses can also, again, like I said, not function on their own like cells can. They don't have enough characteristics of life. Now, if you look this information up on the internet, you may find some information that says that viruses are alive. To some scientists, all viruses really need to have, or anything really needs to have to be considered alive, is genetic material, something that they can pass on. They do reproduce inside of a living thing, but they're still reproducing. So, in the end, you can ultimately make your own decision as to whether viruses are living or not. However, for the educational purposes, I would prefer that you understand how viruses are not living. Viruses are very, very, very small. They're much smaller than bacteria. Their shape, they can be rod-shaped or they can be spherical, generally. They have a protective shell, a capsid, which is the protein coat that I've been mentioning. Some can be covered by an envelope additionally to the capsid. And as I've mentioned already, they have genetic material, which can be DNA, which is double-stranded, or RNA, which is single-stranded. And something interesting is that typically RNA viruses mutate faster and better than DNA viruses. DNA viruses, because they're double-stranded, they also have a mechanism which checks their DNA for mistakes, whereas RNA doesn't, and it's much, much easier for it to change. So here are a couple pictures of some viral shapes. You can see some are very spherical. They also have geometric shapes. And then there are also rod-shaped. The tobacco mosaic virus is rod-shaped. Here you can see a host cell. Excuse my not straight at all line. But, and here you can see the size of a virus. So as you can see, a virus is much, much, much smaller than a cell. Okay, and that's what allows it, makes it so easy for it to get into the cell. But additionally, I wanted to talk a little bit about the envelope. It surrounds the capsid. It could be made of a carb, lipid, protein type structure. And it also helps the virus enter the cell. And some viruses, like bacteriophages, have tail fibers, and this functions like a syringe in allowing the virus to just inject its genetic material into the cell, and the cell takes up the genetic material and starts carrying it out. There are two types of viral reproduction, lytic and lysogenic. In lytic reproduction, the virus injects its genetic material, which uses the cell's organelles and energy to create more viruses, and then eventually explodes out of the host cell, and we sometimes call this virulent. Lysogenic is when the genetic material enters the cell and still attaches to the genetic material, but instead of immediately becoming active, it kind of lays in wait, and the cell continues to reproduce as normal with the viral genetic material. And then days, months, or years later, and even sometimes never, the virus becomes active, enters the lytic cycle, creates more viruses, and then explodes. But we sometimes refer to the lysogenic cycle as temperate. So here's a picture. Starting here, we have the injection of the genetic material into, in this case, a bacterial cell. The genetic material in the lytic cycle becomes active, and it creates more viruses, and then those eventually burst out of the cell. But in the lysogenic cycle, the viral DNA is injected, okay? It becomes part of the cell's DNA. When the cell reproduces, it copies all of the DNA. It does not discriminate between the viral DNA and its own DNA. It's DNA, so it just copies it. Okay? And now we get new cells, but eventually, or again, sometimes never, it becomes active in the cells, creates more viruses, and eventually destroys those cells. An example of a lytic virus is the cold virus. Okay, pretty much within a couple of days of getting the virus, you've got the symptoms. An example of a lysogenic virus is HIV. Sometimes a person can go for years and years and never know that they've got HIV. So it's hard to find a drug that kills the virus and not the host cell. And that's why the discovery of antivirals recently has become a huge step against disease, specifically virus. Okay, we can block viruses from basically functioning in the cell, or maybe even getting into the cell. A new beneficial use is genetic engineering. Scientists have started using viruses to help them. Basically what they do is they take the genetic material out of a virus, the harmful part of the genetic material. They put in a useful genetic material, and then they inject that into a person, and then because that virus is specific, it will inject the genetic material into the cells that need it. And a perfect example of this is cystic fibrosis. So basically they use the cold virus, they destroy it, they take out the harmful genetic material, they insert the gene that the person who has cystic fibrosis is missing, and then they inject that virus into the person. So then that virus delivers the needed gene to all of the cells. The cells take them up, and then those cells become healthier because they now have the genetic material that they used to be missing to make the person have cystic fibrosis. So I took a moment and typed up the information for the NJBCT that the state wants you to know. So take a few minutes, pause this, read through it, see if you understand what the statement is saying, see if you can do what it's asking you to do. Now I'd like you to answer the questions here on loose leaf paper, and I will be collecting this in class tomorrow.